first steps, you get your raw data either from GBS, so your VCF files, or if you have array data, you have to use maybe FitPoly, or there are some other ones you can use that will give you a CSV file that has dosages already called for you. And from that, you do some filtering, and we'll go through each one of these steps and talk about them more in depth. Then after your filtering, you're going to estimate your combination fractions between all the markers. Then you're going to group your markers into chromosomes. Then you have to order your markers. So the very first step is, and if at any time anyone has questions, stop me. Either open your mic or put it in the chat. I have the chat on the second screen, so I'm kind of keeping track of what's going on. So filtering data. So we just, like we talked about last week, we got our GBS data, or in this week's example, we're going to get array-derived data, and you're going to do some filtering. And you might want to filter, and you might want to do some just really fast QC to see if you have any off types. Remember last week we talked about, oh, we might get off types. We could do a simple PCA, and a lot of times you can really quickly tell that you have genotypes that don't belong. Right here on the right is a PCA of the data that have of the two populations that were in the Rose Rosette paper that was in the link that sent out yesterday. And in this PCA, you see kind of these two main groups. And let me see if I can do the laser pointer. Yes, you see these two main groups. Those were our mapping populations, okay? But what do we see? We see that there's this clear off group. They're numbered in such a way that it should have belonged to one of these two groups, but they don't belong. We see that this guy doesn't belong, this guy doesn't belong, and these are supposed to be the parents. The vertices of this triangle are supposed to be the parents, but we have some progeny that cluster with that. So those are most likely maybe selves or just bad data. So at first we have to isolate all these guys, and that's one step of filtering or kind of QC. Other things that we might want to filter on is missing data. So when you get either your VCF or your CSV file that have dosages called, what you'll notice is that for markers on a marker, like across, like on the CSV file, you'll have each row as a marker and each column as a individual. And you can filter on either the rows or the columns and set values for how much missing data you want to allow, either per individual or per marker. Then you can also filter data based off segregation distortion. This is a really good way to first filter your data, because if a marker doesn't fit the expected segregation ratios, then it's probably a bad marker. So that kind of leads me to the first question. Does anybody have any ideas about what are some other things that you can think about that may cause bad data or missing data?
Like bad genotype calls, like erroneous genotyping calls?
Yep. Somebody, Sanjeev just said bad genotyping calls. Richa in the chat says sequencing errors. Kelly said indels in the reference genome, relative to individuals in the population, yes. Those are all good things that could cause, structural, so Alexander says structural variation. So if we were talking about GBS data like last week, it's very easy for our populations to be very different than the reference genome, right? So we can have a lot of missing data there. Maybe minor allele frequency. That sometimes in like GATK or in upstream processes, you might set a either minor allele frequency, or you might set kind of some filter, that downstream becomes NA just because it's a hard filter that was applied very early on. So yeah, those are all good answers.
Can I ask a question now, if
Yep.
it is okay?
Yeah.
You mentioned like filtering data on segregation distortion, yeah?
Mm-hmm.
But segregation distortion is could be like because of biological genuine reasons. So
Yes.
if the markers are clustered together, all
Mm-hmm.
markers which are showing high levels of segregation distortion, if they are following a nice peak, like
Mm-hmm.
initially low significant value and then rising up like a QTL peak,
Mm-hmm.
then does it still make sense to filter those markers out, because these could help rather the linkage mapping?
That's a good question. So in roses are really finicky. I primarily work on roses. In our diploid populations, we do see some instances of where things they don't fit the expected segregation ratios, whether or not that's maybe um, things jumping around, transposons or whatever, that biologically is real and biologically should have that segregation distortion. The problem is a lot of the mapping software don't take segregation distortion into account. So their priors or their assumptions are that things are following Mendelian patterns. So when we include segregation distortion markers, then it's going to result in maps that have large gaps. Now, that's not to say that it's not really there in your population. You might have segregation distortion for real in your population, but there's no real good way to kind of include them. You can play with the p-value of how much segregation distortion you want to allow, but that's the best that I can answer,
Okay,
because
no, thanks. That's very helpful. But when you
yep.
say that segregation distortion is not like factored in this in the program, you are saying for this particular tool which you are using, or
Mm-hmm.
because I think like join map, these tools can, I think, take these kind of markers, but I am not so sure. But when you say this is not included, this is for any linkage mapping software or just for the one you are giving tutorial
For
for?
the ones that I'm aware of, like join map, and for even like MDS, Polymapper, Map Poly, a lot of their
Okay.
assumptions are that uh, things follow normal segregating, because you can't model segregation distortion. It's very difficult to model that. So
Okay.
And
yeah,
the
go
issue
ahead.
with, yes, sorry, Chicken.
Yeah, go ahead.
The issue with segregation distortion is that it's hard to you to distinguish between biological segregation distortion and genotyping errors. So
Yeah, that's why in my question,
so
I don't
maybe,
want to take a lot of time on this question. Just very briefly, that's
yeah,
why I mentioned that when the the markers showing segregation distortion are clustered
Mhm.
together, then it is
yeah,
a good indication that this is because of a biological reason. If there are like odd markers scattered around single mark.
yes. I think that if you can distinguish what's biological and what's segregation, what's genotyping error, the biological segregation distortion should not distort too much your map. Like the map will be built fine, but because we cannot distinguish, probably you have bad maps if you keep the segregation distortion markers there.
Okay, yeah, thanks.
Sergey had a question in the chat. Says, since you are working with polyploid data, is it a clear cutoff that off types may be resulting from selfing? I'm just assuming that these vertices are selves because they cluster very closely in a PCA with the parents, my parental genotypes. That's the only, but there wasn't any cutoff that I was using. I was primarily here just trying to set aside what are actually in my two populations. Okay, I think that is all the questions we have up to now. So next step. So after you filter your data, you get rid of all the a lot of the bad data. You need to estimate the distance or estimate recombination fractions. So the two software that are in Tools for Polyploid grant are MapPoly and PolyMapper, and they take different approaches. So whenever I started working on polyploid genetics 3-4 years ago, PolyMapper was one of the first ones that was published, and so I made some of my first maps using PolyMapper. And what they do is they take all the simplex nullplex markers and calculate the recombination fractions between them. They make a scaffold. So from that step, you have to make your 7 chromosomes or however many chromosomes, and then your homologs from those chromosomes and make a scaffold, and then fit all the other marker types to that. And the pros, I guess, in this strategy is that computationally, you reduce the number of recombination fraction calculations you have to make. Now, more recently, I've been using MapPoly a lot more, just because it's very easy for me to feed it a lot of data and just let it sit there, and it'll go, and I don't have to do very much manipulation. It takes a different approach. It calculates the recombination fractions between all marker types at the very beginning. And when it does this, it requires more RAM, it requires more storage, basically, to keep that object. And as you can kind of think, the more markers that you have, the storage of this object is going to get exponentially larger. They do have, MapPoly is working on and has implemented a quicker way to do this, but you can look at their software and you can find there is a little quicker way to how to estimate all the recombination fractions. But that's the two key differences, is that MapPoly does it between every single marker type, and then with PolyMapper, it does a simplex nullplex first, and then it calculates all the other marker types to those. Let me think. Oh, this picture on the right is a picture generated in MapPoly. You're going to see something similar to this in our tutorial when we go through it in a bit. But what you're seeing is that when markers are very close to each other, in reality, then they're going to have a very low recombination fraction, right? So when you have two markers that are very physically really close together, that are almost linked, you're not going to break that linkage very often. You're only going to break that linkage between those two close markers if you have thousands and thousands and thousands of progeny, right? Now, markers that are very far from each other, let's say on two different ends of the chromosome, are going to have a 0.5 recombination fraction, because on average, a chromosome will break once somewhere on the chromosome, and possibly twice on a chromosome, any given chromosome. So that's why the top end is 0.5, the bottom end is 0.0. And what you see here are the markers ordered in genome order and the recombination fraction matrix. Oh, somebody said, can we share the link for the data set? Yes, I will copy that in there. All right. So what you see here are 7 linkage groups. These are rows linkage groups, 2 being really long. And then you'll see some kind of a pattern of just red through the middle is what you want to see. Now, on really bad data sets, what you won't see is red in the middle. And I've been working with a little bit with GBS data, and GBS data doesn't have as strong of this red. So if you work with GBS data, you'll see a little bit different of a pattern. But this is what we're all striving towards. All right, let me see, next slide. The next step is grouping markers into chromosomes. So after you get your recombination fraction calculations, you can either use that or you can use the genomic data. If you know where your markers are supposed to be, you can use that to group your chromosomes. So MapPoly and PolyMapper also have different approaches to this. MapPoly can either use UPGMA, which is a clustering algorithm, to cluster your 7 groups, and we'll show that in the tutorial in a bit. You can use your genomic data. So if you know which chromosomes your data is from, you can just say, okay, all of these belong to the same chromosome. Or you can use a combination of both. And actually, Gabriel showed me that. So I have this code from a long time ago. And we'll show some of that, and it's code that he wrote from a long time ago. PolyMapper uses a different approach, and we talked about that earlier. It uses all the simplex, nullplex markers to calculate the recombination fraction between those markers, and then you have to make a scaffold, and then you fit those to the scaffold. So I don't think I've tried to use like genomic order or anything in PolyMapper. On whether or not that's possible, I'm not quite sure. But how their vignettes are written, it's not supposed to be done that way. So after you group your markers into linkage groups, then you need to order your markers, or I guess estimate how close the markers are to each other. So MapPoly, you have to give, they use a hidden Markov model chain on a predetermined order. So you have to feed MapPoly either the genomic order, the physical positions on each chromosome of each marker as a given order, or you can feed an MDS order, and it will try to place the markers and fix the, what's that called? Fix the, it will need to, phase all the markers in the right phase. That's what I was thinking about. But you need to provide it a order ahead of time. So that could either be genomic or MDS. PolyMapper takes the groups that you made earlier with the simplex, nullplex markers, and then it takes all the recombination fractions that were calculated in a bunch of steps, and it uses MDS to order the marker. So it uses MDS is written by Christine Hackett, and it's one of the very first like polyploid mapping algorithms. And it basically takes the markers and its recombination fractions, and then it just gets the most likely projection of markers on a curve, and that's where you get the order. So slightly different approaches, but all right, I think that's it. Let's get to the tutorial, the fun part. And this is where a lot of the more of the questions will come up, and people will have problems. But if you have a laptop or a PC that has between 4 to 8 cores, you can probably follow along. Like preferably 16 gigs of RAM, but I think you can get by with 8. This is a pretty small data set. We need MapPoly installed, and we need to download the scripts. So we can show everybody how to install MapPoly. I see that question. So I'm assuming that everybody has R and RStudio. So hopefully everybody has R and RStudio. So go ahead and pull that up. So if y'all went to this GitHub and y'all downloaded the zip, the file that we're going to look at, oh wait, this is slow because I guess everybody's trying to download it. Okay, it's downloaded. You're going to extract the zip. And inside the zip, the data that we need is in data, and it's this guy. And the tutorial we're going to follow is a mapping example. And when you open that in R, you're going to get something that looks like this. So I'm going to go back to my version that I had. So let's test this in the, I guess in the Zoom app. If you have RStudio pulled up and you have opened up the tutorial RMD, can you give a thumbs up emoji? Yeah, we got 1, 2. Got 2 people following along. 3, 4. Oh, someone says they're still installing. And those of y'all that have this pulled up, make sure you have Map Poly installed. And you'll know if you have Map Poly installed if in the console you say library Map Poly. And if it doesn't give you an error, then you have Map Poly installed. Can everybody see the text on this, or do I need to make it larger? Is this fine?
Can you make a little bit larger, please?
All right, make it a little larger. Is that good? All right. Are people starting to get Map Poly installed? All right, somebody says it's installed. Can people, yeah, people have opened this up, right? Yeah, people, someone saying in the chat it's taking a little while to install Map Poly. Yeah. Can you show the link again? Yes, I can show the link again. The link to the GitHub, or it's a good question, Chris. Can you help me with Cesar's question?
Yes, I think in the last session I was telling about how to do the genotype calling and the SNP calling to try to avoid the errors. The issue with the GPS is that you have much more genotyping error than the array, at least the ones that we have here. Like with our GPS is not like this recombination fraction that GCAN did. So we have many strategies to filter the markers before going to this stage. So yes, I can share with you some of our works that explain a little bit of the filters that you can use in your data too. I will share here in the chat.
Maybe I should show them of what kind of recombination fraction matrix we get in GBS, if I can find one. I'm sure we have one in.
Yes, and like I showed last week the reads to map workflows, and it was all developed to try to figure out how to do the filter in GBS and how to improve it. So I was about to share its paper because there I suggested some things that worked for our data set and for the diploids, and we already did this for polyploids too, so we know that it will work for polyploids. So I can share with you.
This is an example of like 2 chromosomes. Like it's definitely not as good as the array, just because of how much missing data there. But you can kind of see between the 2 chromosomes, that between the chromosomes they're not linked, but there's some semblance of red in there. That's just an example of what GBS might look like. All right, so does most people have MapPoly installed? We're good? People that want to follow along? All right, so what we're going to do is import the data, and it's going to be called BEMG subset. It's going to be on a different place on your computer. So if you downloaded everything as a zip, right? And you unzipped the folder and you're inside it, make your working directory where this thing is, this mapping example RMD, by going to session, setting the working directory, and to source file location. If you click that, it'll put you wherever your folder is inside of the tutorial folder. And then everything should work by just clicking. It should. All right. So if you've got it set up, we're good. See if you can make this first chunk of code run by clicking this little run button. If you click that, it imports the data. And it gives us a little printout. And then what it tells us is that we have a data set that has a Ploidy of 4. There are 161 individuals in this data set. There's 1,382 markers. All of these markers are informative, and the only reason why the number of informative markers is so high is this is a very good subset of data set that will run on small PCs. And it does some filtering in the background. All right, so everybody done this first step? Get a thumbs up. All right, all right, all right. Nobody's stuck yet, I don't think. The RMD file is inside of tutorial. Oh, somebody said they're stuck. Oh, there's multiple people that are stuck. All right, so the RMD file is in this tutorial folder. So after you download the zip from GitHub, you can install PolyMapR if you want, but we're not going to explicitly use it. There's one step that we will use it for 1 or 2 of its functions. So you can go ahead and install PolyMapR if you, at the very end, we'll show an example of things that it can do that helps kind of pre-processing too. What kind of error are you getting in MapPoly? I don't know if maybe Gabriel can help Chris. All right, so I'm assuming they found the RMD. Okay, keep going. Now, last week we talked about, so this file that we just imported into this DAT object is a CSV, is a CSV that was generated from array data that was called in FitPoly, and then it spit out this thing, and we put it in a format that MapPoly can read. And that format looks like this. There's a picture right here as a marker, as the P1 and P2 dosage. It has the linkage group it belongs to, the position, the genomic position, and it has the dosage for all the individuals. So this is what it looks like. Now, just as a demonstration, we can also input VCFs or read VCFs, generally are what come out of the GBS pipeline. And if you run this line, it'll show you how it does it. It takes a little bit and it'll read it in, but we're not going to actually use this line. So it takes a while and then finally goes. Now, typically your next step would be filtering your data. And because this is such a clean data set, you're not going to really filter anything out. You can still run this line. It's not going to really do anything. Just kind of as a guideline, the tutorials and the vignettes suggest around maybe 5% or 10% just to start out, just to see what you get. And a lot of these functions have graphics that kind of show you how many markers you're throwing out. So for example, let me run this. If INTR is true, I'll run it down here so it actually, what it'll do is it'll give you pictures of how many markers you're filtering out. This might slightly look different from the version that y'all have from CRAN because I'm using the most updated version of MapPoly, and I think Marcelo added new graphics to how many markers are filtered out. But this is, so what we could do is we could down here say, I could put 0.01. Why is it not letting me type 0.01? And what I'll do is it'll filter out 600 of the markers, but I don't want to do that. So I'm going to press N. So our data object still has 161 individuals and still 1382 markers. All right. So we're going to run this step anyways, where it's not going to really filter out much. And in this output, nothing gets filtered out. Still same amount of individuals. So real world use, you'll have a lot of markers being filtered out. But for this exercise, we don't have very much. So the next step is filtering by segregation distortion. So there's going to be very few markers actually removed. There are a few, but what we see here in this little chunk of code is we're using a Bonferroni correction for our cutoff for the segregation distortion. So we're looking at a p-value of 0.5 divided by the number of markers. So this DAT object, which is a MapPoly data object, has in here the number of markers as one of its, I guess, one of its sub-objects in there. It has a number of markers. So it's 0.05 divided by the number of markers. It's a lot smaller of a number. And then this chunk of code will filter based off segregation distortion on this data object using the Bonferroni corrected p-value. And this also has like an interactive way to move your p-value if you want. But for the purposes of this, we're not going to do it. We're just going to run this. And it'll give you a pretty little plot. Actually, it gives in your R console, it tells you that now your data set has a little bit fewer markers. Now it has 1369 markers. So it kicked out a few. And it gives you this plot. This plot might not look like y'all's. The color scheme's a little bit different because Marcelo did some changes in the color scheme than the version that's on CRAN. And if y'all are familiar with DevTools, you could get the newest version if you want, but it's not as stable as the one that's on CRAN. But what you see here is a matrix of the genotypes. So on this axis, you have the individuals. And on this axis, you have the markers. And the different colors are the different genotypic calls or dosages. On the top, you have the segregation distortion p-values. And then on this side, you have what kind of markers are in this data set. What kind of segregation is in this data set. So this step will take maybe 2 or 3 minutes. So we're going to go ahead and execute this box. Just to be careful, before you run this box, we need to know how many cores your computer has. So I think it's parallel detect cores. So if you run this line, I'll put it in the chat. If you run this line down in the console, it'll tell you how many cores you have available on your computer. Don't go over that. So in my case, I'm just going to use 6. And I'm going to click run. So this will take a few minutes, but does anybody have any questions yet? People that are following along. Look in the chat. Seems like Chris is answering questions. Gabriel's helping install. Oh, somebody asked, Bonferroni looks to be too stringent with my data. Is there a way to use FDR? The FDR and the Bonferroni corrections are going to be very similarly small, I think. But there's no way built into MapPoly. You can just manually look at the cutoff. And when you manually, if you do the INTER as true, you'll see kind of a cutoff of what's that called? You'll get like a plot, and you can kind of move that plot up and down, change your p-value so you don't throw out too many markers or you don't retain too many markers, and kind of play around with that. So I hope that kind of answers your question. Somebody asked, is there a minimum or maximum threshold for number of markers? Good question. For MapPoly, the more markers you use, the longer this estimate pairwise recombination fraction step is going to take, because it's making, so marker 1 is going to be compared to marker 2, marker 3, marker 4, marker 5, so on and so forth. And it's going to do that for the second marker, and it's going to do that for all the markers. So in this output, it's telling me that it made 900,000, roughly 900,000 pairs of markers, like comparisons, and it has to store the object somewhere, and it's storing this object here in this up here, this all RF pairwise object. And the larger, the more markers that you have going into this step, the longer it's going to take, and this object can get really big. So hopefully this step is starting the end for people that are following along. If you've finished this step, can you do a thumbs up? All right, Tessa's done. Falcon's
Does
done,
does
okay.
this program also take into account the SNP locations, only those locations which are present in the parents, to avoid any other erroneous genotyping calls in the progeny?
So it depends if you can choose that. For MapPoly, you can choose whether or not to use the genomic information if you want. You can choose to just use grouping based on, which is the next step, and just use the grouping algorithms. And you might be able to get it to group, you might not, just depending on how clean your data is.
No, I
But
meant like if only include the SNP positions if those are present in the parents.
You can't have, so you have to have the parental call to have the dosage in the parents. Yeah, well, you can't have any missing data in the parents. P1 or P2 can't be missing. Does that answer your question?
No, I was just saying to avoid the SNP calls which could be due to errors. Just take into account only those positions or SNP locations which are present in the parents.
I'm not quite sure I understand the question. If anybody else, if Chris or Gabriel understands the question better, or maybe I'm just not understanding it.
Just to avoid the like genotyping errors or wrong SNPs which are called due to any genotyping or sequencing issue, just to take into account those SNP locations which are present in the parents, and then any other locations will be like filtered out.
I mean, you could do
Or-
that, but that's way further upstream than
Okay.
than here. You would do that before you even get into the mapping.
Yeah, that's
You
what I
would
was
choose-
asking, if the
Yeah, yeah, yeah.
software can take into account that information.
Yes, I don't know if I'm wrong. Correct me, Gabriel, if I say something wrong, but I think MapPoly already filter markers that are missing in the parents. When you go into the software with your data, if you have missing data in the parents, MapPoly will discard the marker.
Exactly.
that's
okay,
correct.
Thanks. That's exactly what I was asking. Thanks. That's
Mhm.
great. Yeah. Cheers.
So if it's a genotype error, the error needed to be happen on both sides, and then that chance, that probability should be low to me. Or in the progeny.
So I don't know if I've understood, but the parents' genotypes, they would like it makes the marker type. So you will have like a single dose, double dose according with the parents. And this you can't put a probability for that in MapPoly, but you can't put a probability in the progen genotypes. So if you know that some genotypes in the progen have more error than the others, you can, instead of input as a qualitative genotype, you can put a quantitative probability.
Yes, I agree, and I'm thinking Sanjay will mention whether in the progeny, whether that is genotype error or is a real recombination. If I understand him correctly, no.
No. Thanks. My question is answered by. Yeah.
Okay, thank you.
Because I was just saying that only take only those markers which are validated in parents.
Mm-hmm.
Only take those markers forward for mapping. That's what, and then that question is answered. Yeah, thanks.
Yeah.
Mm-hmm. And so because we cannot give a probability for the parents, it's important that the parents, if you're dealing like sequencing data, or it's important you to repeat the sample and have like higher depth or higher quality in the parents, because if they are wrong, we'll be all wrong. So it's important
Yeah,
that the
yeah,
parents is better.
exactly. That's why what we tend to do, like sequence parents at higher read depth have more robust genotyping calls, and then
Mhm.
just take those positions for the linkage mapping. Yeah.
Okay.
All right. So the next step is to run this line 93, this box. And this is where we get that plot of the recombination fraction matrix. And it shouldn't take too long. I think everybody should be caught up to this point. And you get this pretty matrix. So the goal next is to try to get all your markers into groups. And there's different strategies for that. And we're going to actually run all three strategies. So this first strategy is using UPGMA. And if you just run this chunk, it outputs a few different things. In our console, you'll see the groups that it's grouped in, the number of markers are grouped in. You see a few that aren't grouped with is genomic order. You also get an output of all the markers into, this is something more familiar that you would see, I guess in JoinMap does something similar where you see all the groups kind of where they fall out. This is similar. And this data frame just shows how many markers are in each group. That line should run really quickly. So UPGMA is just a grouping algorithm that takes the information of the recombination fractions matrix, and it will group things on the things that are more similar to each other. Now you can make the groups using other methods. So if you run this next box, we'll look at the output. So first I'm making a group that is the intersect of our genomic information. So that CSV that we put in, it had linkage groups, it had where the position of linkage group, that's the genomic information. So over here, we're making groups that is the intersect between what we found in UPGMA from the recombination fractions and the genomic information, where these SNPs are located in the genome. We can only look at the genomic order. We could just group it. Okay, maybe I trust my my SNP calling, or I trust where my chip SNP chip blasted areas are on the genome. Maybe I trust that genomic information more. Then you could use the genomic information. Or maybe you don't have any genomic information. Maybe you're working on a crop that only has scaffolds, or it only has, or may not even have a reference genome. Then you would have to probably use UPGMA. So when we run that line of code, this output right here, this is thanks to Gabriel. What it does, it tells you how many markers are in each group. So this first group is intersect of UPGMA and the genomic order. And this is for chromosomes 1 through 7. And then this is looking at just the genomic, and this is just UPGMA. All right. So genomic is supposed to have more, but it looks like UPGMA groups some things that aren't really in the genomic group. But typically on a full data set, you're going to see, I think, genomic probably have more. So depending on how you want to proceed, you can pick any of these groupings. So for the purposes of this tutorial, we're going to take the intersect of UPGMA and the genomic information, and we're going to assign it to an object called LGS. So we need to run this line. So right now, we've taken our raw data, we've filtered it, we've estimated recombination fractions. We've grouped our markers, and now we need to have an order. So we're going to run the HMM chain using the genomic order that was provided. And we're going to run it on one chromosome so you can see what the chain is doing. And then the next step is we're going to run them on all 7 chromosomes. So if you run this block of code, you're going to start seeing things run. You're going to see things output. And this is the chain trying to phase the markers in the right place, in the right phase. And this will run relatively quickly. It'll be maybe 2 or 3 minutes. So what this function does is it takes a input sequence, and then these can be determined. These are parameters of the HMM chain. And you can be more strict or you could be more lenient. And you have to provide it a recombination fraction object, which is the 2-point comparison between all the markers in this group. And this is also the chain's parameters. So when it's done, it tells you in this, I think this was linkage group 1, that it initially had 155 markers in this group, and it could map 154 of those markers, and it kicked out 1 because it couldn't fit its phase. And we see that it is marker number 13 that was kicked out. So that's what it's doing in the background. Now, if we want to look at this one chromosome, line 180, if you run this block of code, you can see this is a graphical representation of what was just mapped. So you have parent 1 and parent 2. You have what dose or the parent 1 and parent 2 was, and these are all phased now. Now, because this next step is going to take a little longer, we're going to have questions after the next step. Go ahead and press go on this next chunk of code on 186. Now, there's a caveat. Before you start, I don't know, does anybody have less than 8 cores on their computer? Does everybody at least have 8 cores that's following along? Remember when we did the parallel detect cores? Because we're going to need to change it if you have less than 8 cores. And it's going to take a little longer. Okay, it looks like everybody's okay. Oh, somebody has less than 8 cores. All right, if you have less than 8 cores, come down to line 215 and put this. If the person that has less than 8 cores, do you have 4 cores or 6 cores, or it's going to take you a little longer? So you have 4 cores. I would just change this to a 3 so your computer doesn't get like really slowed down. So change line 215 to 3 here and make cluster. And also in line 223, change that to a 3. Otherwise, those of you that have 8 cores, just run the code as is. For Benny that says they had 4 cores, this step is going to take for you maybe 3 times as long, just because there's 7 chromosomes. We're mapping all the chromosomes at once. So go ahead and click this, and we'll just wait, and it's going to run. And we can stop to have questions. Are people kind of following along? Questions, comments? You don't have to put them in the chat. You can talk out loud too.
I have a question
What
regarding grouping. Hello?
Yep.
Yeah. Can you hear me?
Yes, I can hear you.
Yeah, I have a question regarding grouping. Like you give expected groups 7. So is there any way we can? I mean, if we don't know how many groups we have, like based on LOD or something, we can.
Oh, that's a good question.
Yeah.
That might be a Gabriel question. So Gabriel works closer with Marcelo than I do. Down the hall.
Yeah. So actually, I haven't seen a situation like that where you don't know the number of linkage groups you expect. So that's why we implemented this way. But it might be possible to try to run this with different numbers and see which one fits better with the data based on the recombination fractions. Other thing you can try to do is calculate the recombination fraction matrix and try to account the number of diagonal blocks you can find in the whole matrix, considering all the chromosomes, and that might give you an idea on the number of linkage groups you have in our species. I don't know if Chris have an idea on that. So that's what I would do in this situation. Yeah.
Yes, I would do the same. Like just seeing the blocks.
Yeah.
Okay. Yeah. Like for example, I work with the sugarcane, so they have like 150, 200 linkage groups, and also, I mean, looking at plot might not be like a viable option as well. No?
Yeah. So yeah, in this situation, especially if you have a lower number of markers, I would try to get this information a priori with other techniques, like using other molecular techniques, other wet lab techniques. Yeah.
But yeah, that's the only option we have. Expected groups should be given. Okay. Yeah. Thank you.
Does Sergey have a question? He has a raised hand.
Yeah. If you genotype data include markers that were not mapped on the genome, is there a strategy to include them?
I'm sorry, can you repeat the question? You were cutting in and out.
If you genotype data include markers on map, is there a strategy to include them and place them on the genome, on the map? If you have on-map markers in your genotype data.
Yeah, Gabriel, MapPoly has something like Trisec that you can include a single marker.
As far as I know, we haven't implemented this in a function, but we have used this approach. So if anyone needs this code, we can just send you by email or even think on implementing this as a function. Yeah.
So I understood that he doesn't have like all markers in the chromosome, the sequence, the genome sequence of the chromosome. He has some markers in scaffolds yet, and he wants to include in the map.
Exactly.
And so MapPoly has an approach for that.
Okay.
Yeah. Yeah. So yeah, in this situation, you can think on 2 approaches. The first one would be running the RF and the MDS, which would try to find the best position of these markers in the chromosome inside the linkage groups actually. And the other one would be trying to run this try algorithm, which Chris mentioned. And we have the code for that. If anyone wants, we can share. Just send us a message.
That would be without giving the pre-ordering.
Yes.
In the map. Okay.
Yeah. Those 2 approaches, you trust on the RF and the data to find the best order. Yeah.
But the try algorithm will take a very long time.
Yes.
Yeah, definitely. Yeah.
So one thing to clarify, this mapping is biparental based mapping, not like GWAS panel mapping.
Yes, this is biparental mapping, and at the very end, we're going to have genotypic probabilities of each parental homolog.
Okay, so anybody know both alpha from the rows can comment how they are different or how they are same or will this process the same for alfalfa? Alfalfa is all allotetraploid also.
Oh,
Anybody
alfalfa
know?
is allotetraploid?
Yeah.
If it's
Oh
allotetraploid,
no, actually, auto.
oh, it's auto.
Yeah.
Is alfalfa bred as a synthetic, or is it? Do y'all have mapping populations or?
Well, we have inbred depression,
Okay.
and my method is we use F1 as a mapping population.
Mhm.
Not like a. I'm not sure the rows. That's why I'm asking
Oh,
this
I see your question. I
Yeah.
see your question. Yes, this is a F1 mapping population.
This is
Roses are highly heterozygous and they outcross.
okay. So that's
They tolerate some inbreeding, but this is a F1 population.
I see. Does anybody working has experience on both alfalfa and rose? Can comment on the difference between the two crops as to mapping?
I haven't worked on alfalfa.
Okay.
So my chunk of code is done. So people's will start being done, I think, soon.
So one more question is,
Yep.
if I understand the situation correctly, the calculation recombination frequency and order of the marker is very important. How tolerant the program to incorrectly order the markers?
Incorrectly order the markers.
Yeah. For example, for the recombination frequency property estimated correctly, you put marker 1, marker 2, marker 100, and two of the markers, like marker 2, marker X and Y, they should be neighbor, but they're the two switched. What is the consequence for the wrong order of the markers?
So for map poly, how it's written, if you use it like it's supposed to be used, what will happen is if markers that you have to give it a sequence. So an a priori sequence first, and then you give it a 2-point, and it tries to put it in that order. And if it can't put it in that order, and it doesn't match the phase, it's going to kick out that marker.
Okay, so
and it won't be included.
Yeah, you are thinking the probability the two markers are incorrectly ordered is really, really low. Or the no chance.
That's so right now, a lot of the ways that we go is we use the physical order. So we're assuming that the physical order is a good approximation of what is reality. Okay. But you have issues like for us on rows, if you go look at the paper, on chromosome 3, there's an inversion because the reference genome that we use has a little section that is not in the same orientation as our germplasm. So if there's issues where there's inversions, maybe in that area, the chain is going to kick out more markers because it can't fit the phases. It can't find the right phase. So it's a possibility that you'll get things thrown out.
Thank you.
Cesar has 2 questions. Or he might be typing them out.
Cesar, can you hear us?
Cesar might be, it says, with this parallel function, I will change the object LGS.
Hi, sorry, this is me. I think that this is in summary that could be the same process because I won't changing the LGS object comes from the second part, but I just need the first part, the first analysis, right? GRS comes from grouping, the first part, using UPGMA, right? The
Yes.
first matrix. The second one is to repeat the same process, using a second approach, right?
Yes. Yes.
So, and the second part is with this map object is all that I need to import into Polymap. Yes.
Oh, so you'll need, so with the map at the very end, when we get a map,
Yes,
your question here, if you wanted to use the genomic, you would just use the LGS genomic, and then you keep running the rest of the script if you wanted to. I just use this temporary LGS kind of, so I don't have to keep changing the rest of the code. I just reuse a lot of my code.
Perfect.
So just whatever you want to input, you could use the genomic order, you could use UPGMA, you can use the intersect. We just happen to use the intersect with this data set. And then at the very end, when you get a maps, the MAPS object or whatever you assign it to, we will have to calculate some genotypic probabilities before we can go into QTL mapping, into QTL poly.
Yes, this is a list of this object map is a list with,
yes. So,
in this case, 7 elements, one
yes.
for each chromosome.
Yes.
And this is in this case is the object that is the mapping. But in the case that I am looking for a QTL, this is the object that I am using to export to other formats without filtering.
Yes and no. So you're going to have to do more. So are most people caught up? Have people run that huge chunk of code? Thumbs up. Their computer's done. All right. So I see some people are done. Let me run this chunk first. I'll get to your question in a bit, Caesar. But this is just a plot of on the x-axis is the genomic position. On the y-axis is the centimorgans. And all we see here is that it follows. There's no major gaps. And you also get a printout if when you do summary maps. You get a summary of your maps. You get map length per chromosome. You get totals, the number of each kind of markers. You get the maximum gap that we see. So to go to Caesar's question, so now you have a map. What do? We have to calculate another object that has the probabilities that each individual carries a specific homolog. So we have to use that object to go into QTL mapping. First we need to calculate, we can run this line. I think it'll take a little bit. And this is what these objects that we're saving, we're going to use next week, I believe. I think Gabriel is going to keep using this data set, and then we're going to use the data set that's in the paper to look for that one strong QTL on linkage group 5. And we're going to use the objects that we generate this week, next week. So this block of code is one of the things that we need. And what this is it is calculating, it's a genotypic probability object that carries, how do I explain it, Gabriel? It carries all the probabilities of the individuals having the parental homolog, basically.
Yes. So for this case, you have 36 possible genotypes for each position on the genome. So it's basically an array with individuals, markers, and all the 36 possible genotypes and their associated probabilities. So yeah, that's it.
Yep. And then we assign it a global error here of 0.5. We'll see a little graphic later on of what happens when we give it a global error. When we give it a global error, we let the chain be more flexible to visit other states and visit other possible phases to get hopefully a better map. And then there's a second calculation that we need here called calculate homolog probability. And this is what gives us the pictures of well, let me go ahead and run it, and let me go grab the code to print out one of the individuals because I forgot to put that in the vignette. All of this is in a more detailed vignette, and I'm going to link that. This is from last year's. This is a more detailed vignette. If you go to Matt Poly's GitHub, just wanted to make it more broken down to show you the basic steps, so we're not here all day. Let me see if I can find. So if you already calculated the homolog probabilities, we can plot, for instance, linkage group 1, individual 10, and that will give us this plot right here. So for this individual, it is carrying these color shaded bars are the probability that this individual on linkage group 1 is carrying the parental homologs of A. This one is about 70-something% that's carrying B. This one's carrying the entire homolog of C, so on and so forth. So this is a visual representation of basically the probabilities we calculated up here. That's used for QTL mapping. So in QTL mapping, what it does, well, what Gabriel will talk about next week is it looks at each position and then it looks at the phenotype. And I see it says, okay, does this specific homolog at this part attribute anything to this phenotype? Yes or no? And it does a bunch of statistics in the background to figure out which homolog or homologs are responsible for a certain trait by looking at all the individuals and their probabilities, their parental probabilities. Does that make sense? Does this graphic make sense?
Well, not sure I understand this one. So
Yep.
that means here, under this figure, this is a linkage group 1.
Yep.
And you will calculate each marker. They could go to different homologs group. Then you have 7 linkage group or 7 chromosomes. So each marker will have 7 probability, and then we'll go to we'll assign the marker with the highest probability to go into one of the 7 chromosomes.
No. So this is after mapping. After mapping, we have these are the probability that this individual inherited the parental homologs. So which version? So because this is an autotetraploid, right? Autotetraploid P1, P2 P1 is donating 2 chromosomes or 2 homologs. P2 is also donating 2, but we don't know which 2. So after the HMM chain is run, you can, it calculates the probability that each individual carries which parental chunk.
So then
this
each individual will have two probability, either coming from father or coming from mother?
Yes, it'll each have, at each position, you're going to have 4. 2 coming from each.
Okay, yeah.
We're just trying to figure out, oh, does it have the E homolog or the F homolog from P2, or does it have the G or the H? So if you go to another individual, let's go to individual 1, when we run this, it's going to look a lot different.
So
And
under
here-
the example, so this marker will go to which homologous?
So this individual at marker, so the x-axis is the map position. And each map position, these are a bunch of markers under here. So let's say at centimorgan 0, at the very beginning of the map, this individual is carrying the H homolog and the F homolog from parent 2, and it's carrying the C homolog and the D homolog from parent 1.
Okay,
And then when you get to position,
different marker,
right at this position, around 18, there was a, what's that called? A recombination event. So it did carry the C homolog up to about 18, and then it started carrying, there was a recombination, there was a crossover. So now it's carrying the A homolog.
So this is a whole linkage group. Then what is X16035? What's that number for? The name X16035.
This is a progeny. This is one progeny.
Okay.
So this is
Gotcha.
one individual in the mapping population. This is one linkage group.
Yeah.
This is the linkage group centimorgan position, the x-axis,
Okay.
and then the y-axis is the probability that it carries any one of those homologs. So all the breakpoints are where we have our crossover events.
So from this map, the breakpoints is around 30 meg.
Yeah. You have one here, you have
Uh-huh.
a breakpoint here, and you have a breakpoint here.
Yeah. Yeah, makes sense right now. Thank you.
Yep. So let me think. Let me see if there's much more after this. Oh yeah, there is. There's a few more after this. So say for instance, you want to, maybe you were like me 3 years ago and don't have access to high performance computing, and you need Polymapper. You might need, because it requires less computational resources because of the strategy that it takes. Now, Polymapper doesn't take VCFs natively, but the cool thing about the tools for Polyploid grant is that all the developers are friendly towards each other and have conversion tools built into each other's packages. So if you had a VCF but you don't have the high performance computing resources to map, and you need to use Polymapper, what you can do is you can take the VCF, you can import it into MapPoly and tell MapPoly to export into a format that Polymapper can use. So we're going to do that as an exercise. We're going to take the data that we have, even though it's chip data, we can still export this DAT object. We're going to export it to Polymapper. This is a MapPoly function. And we're going to apply some things that Polymapper can do that MapPoly doesn't have built into it. So when you run this chunk, we exported the data into a object called all dosages. It's just a matrix. And I'm screening. So in Polymapper, it has a function really early on where it screens for duplicate individuals. Because a lot of times in experiments, what will happen is you're going to mislabel tubes, you're going to mislabel plants, and it's very possible that you have the same individual with different names. And what Polymapper did was, in this example that we just carried through from the very top to now, we had 4 duplicates. So we had actually 4 individuals that were duplicates of other individuals in this data set. So a best practice would have been to run this first before we even did the mapping, right? We had to find which the duplicates are, either merge the duplicates or throw away the duplicates. So that's one function that it has. Another function that Polymapper has that that's not built into MapPoly is the PCA progeny. And this just shows a PCA of all your progeny, and your parents should in this plot be in the middle. And I didn't highlight P1 and P2, but they should be somewhere in the middle. Let me think, what else? Yeah, so those are 2 things that we should have done early on, just as normal QC. So we have this other data set in the data folder called BE subset remove duplicate. And this is with the duplicates removed. And if you're curious, you can run this chunk of code. It's not going to give you that much difference. But if you want to run that chunk of code, you can run it. If not, let me see. I think I have, if you go to the tutorial box and you click mapping example HTML, it has everything already run for you. So I'll show you the difference, so we don't have to run it. Where is it? Where is it? Where is it? Where is it? Yeah, it's after this. We run the map, taking out the individuals. So this is the summary, the 2 maps. So we have the map up here is the one that we originally did. And this is the one after. Somebody said, can we reshare the files and codes, please? Yes, they are on this GitHub that I'll send in a link. And if you just want to go to this HTML file that we show, if you click on this link, it'll take you there. But what you see here is that there's not that much difference. It's about the same. We can look at the summary of the 2 maps. This is the one before, and this is after taking out the duplicates. You slightly get a slightly smaller map, 519 total instead of 520 total. So virtually they're the same map. Those 4 individuals didn't do too much damage. Now, next week, we're going to take output from actually that last, if you want to carry on your computer for next week, you're going to have to run that big chunk of code and get this object. It will be saved in that folder, and it will be used next week. However, I think we can also just provide the object, so you don't have to run it, and that would be easier. Let me see. Oh, there's one more cool graphic that I want to show y'all and kind of explain. If I can get to it. And I don't want to run it, so I can just show y'all. Come on, computer. This is not supposed to be blurry. Guess there's too many things running on my computer. Oh, there. Ah, where'd it go? There. So this is a graphic. I don't know if y'all can see this very well, but this is similar to the graphic that you see at the very beginning of map poly, where the x-axis is the markers, but now these are the markers in order of the HMM chain. It's the map order of all 7 chromosomes. On the y-axis is the individuals, and all the black is unchanged genotype dosages. So the chain will take all your data, it'll run, and then it'll make small changes in the dosages to be able to fit a map. And this is actually hidden inside of a bunch of objects inside of map poly, and you have to go and look for it. But what this is showing is that before you do any kind of global error on the left, what happens is all these green dots are imputed data. So a lot of things that were made missing at the very beginning, the chain is able to impute that data and give it a genotypic dosage. There are a few red dots in here, and that is when the dosages that were changed after the HMM chain doesn't match the input dosage that was given, that CSV file that we gave it at the very beginning. Now, over here on the right, this is a picture of after you run the chain, you can give it a global error. And when you give something a global error, what it does is it allows a chain to make more changes, or visit more states and make more changes in the genotype dosage. And we see the same thing. The greens don't change. The imputed is the same. But the red, you get a little bit more, get up to about 0.5% of all the genotype. That is something that changed. So maybe a dosage from 1 changed to a 3, maybe 2 changed to a 1 or something like that. So you allow the chain to make more changes so it makes a better map. So whenever you apply a global error, what happens is you see the map shrinks, it gets smaller because it's making these tiny changes in the back to the original data. So I hope that wasn't all just a mumbo jumbo mess. Hope that sort of made sense. Does anybody have any more questions? If you have technical questions, you should ask it, because we have Gabriel here. He understands this a whole lot better than I do.
I have a question I not feel understand completely compare with the alpha and the rows. So when we talk about allele frequency, let's say we have 100 progenies. And if we have the resistant allele and susceptible allele, if we have 100 progeny, 90 of those susceptible, 10 of those resistant. So the resistant allele frequency is 10%.
Mm-hmm.
And but when we talk about dosage, my understanding, we talk a dosage like one gene, one progeny. You have allele like AA, 3A, 1G, or 2A, 2G. Those are dosage. It's the same concept as I understand for alpha and the rows. What can somebody know both the two crops, and is the definition of dosage, allele dosage and allele frequency are the same between the two species?
No, allele frequency is how often you see the reference allele or the alternative allele showing up in your population. But dosage is the number of, so in an autotetraploid, you're going, at any locus, you're going to have 4 homologs. Dosage is the number of alternative alleles at any given marker.
So dosage is for individuals. And
yes.
frequency is for population.
Population wide.
Yeah.